Okay, here's our introduction to the urinalysis laboratory. And so I just want to remind you at this point that you don't want to empty your bladder before coming to class, or if you do have to empty your bladder, make sure to run to class, pick up a cup, and take it to the bathroom with you so you can fill the cup and have it ready to go when we start lab. All right, so we're going to be starting this lesson with a little bit of anatomy, and we're going to be starting it with the cardiovascular anatomy, which we did a couple of weeks ago, but we're going to be doing it again because it's actually pretty salient for our discussion of the urinalysis laboratory. So bear with me. So just reminding you that we have the heart. The heart has a right ventricle and a left ventricle, RV, LV, and then the RV is filling with blood from the right atrium. The left ventricle is filling with blood from the left atrium, and the general circulation is like this. We have blood returning back to the heart, basically blood returning back to the heart. It's coming back in to this right atrium from the superior vena cava, that's this part over here. The inferior vena cava is down over here. The right atrium fills with blood from that returning blood. The blood goes down to the right ventricle, then it goes out till the pulmonary circuit. Okay? Now we're not going to be paying attention to the pulmonary circuit because we're focusing our attention here on those parts of the heart that are pertinent to the urinary system. Okay? And then the oxygenated blood returns back into the left atrium from the pulmonary veins, flows into the left ventricle, and then it gets pumped out. And here's where we want to kind of remind ourselves of what the whole structure looks like, because we've got this big old artery. It's the aortic arch, and it comes up. Okay, this is the artery. It actually pokes up from the front of the heart, comes out like this, and it makes a big arch, and then it flows backwards. And then the flow of blood kind of arches backward like this. So we actually end up seeing the aorta, the descending aorta, going down the body in this direction, carrying the oxygenated blood to the entire body. You should remember at this point that the blood returning back to the heart through the right atrium, through the inferior and superior vena cava, it's going to be deoxygenated blood. It flows down, and basically the blood returning back to the heart is going to be coming back with a full charge of oxygen, and this returning blood from the pulmonary circuit is going to be coming into the left ventricle, and from the left ventricle it's going to be pumped upward like this into the arterial system. Okay? And along the way, it's going to be, ultimately, this blood is going to be passing downwards, downwards, downwards. This is the same blood that goes down the trunk of your body, down to the legs. Eventually, it splits into the femorals down here. But before it does that, we've got a couple of arteries coming down. We've got the right coronary artery. We've got the left coronary artery. We've got the right, this is the right subclavian. Basically, also connects to the right common carotid. Then we've got the left common carotid. The carotids are the blood vessels that take the blood out to up to the head, up through your neck. This would be the artery that serves your right arm. Then you've got another artery serving your left arm over here, and so we've got the arms and the heads served. We've got the heart itself served, and then if we follow this down far enough, okay, this is the aorta. It's going to eventually head down to the legs, but before it gets there, we're going to have the blood supplied to the kidneys. So this is going to be the renal arteries. The right renal artery and the left renal artery. Okay. The blood from the kidneys, the deoxygenated blood from the kidneys after the kidneys have done their work, will have clean and deoxygenated blood. Blood that's been cleaned because it's been through the kidney, but also deoxygenated blood because it served oxygen to all the kidney tissues. It's going to be returning blood back to the heart through the inferior vena cava. So here's the vein, and so the kidneys are going to be returning blood through the renal vein, and so the right kidney, the left kidney is over here. It's, the blood returning back to that kidney is going to be coming back to the renal vein. Okay, so let me draw that a little bit better. Renal vein is going to be taking deoxygenated and clean blood back into the renal vein. It's going to be taking it back up to the heart. Renal artery is going to be carrying oxygenated blood to the kidneys, and basically it's going to be coming out into the renal arterioles. We'll be talking about those later. Same thing on the other kidney. The other kidney is going to be receiving oxygenated blood from the heart through the renal artery. The blood is going to be going out to the renal arterioles, basically going out to the nephri, and the deoxygenated blood that has also been cleaned is going to be returning back to the renal venules. It's going to come back to the renal vein over here, and it's going to be returning that blood back up to the heart. So basically, circulation out to the kidneys is going to be returning blood that's going to be entirely clean. Now, the other thing that's going to be happening is that all the impurities from the blood are going to be collected in urine. And the urine produced by the kidneys is going to be going to the urinary bladder, which is down here someplace. So if this is your urinary bladder, there's a urethra, and that's going to be avoiding the urine. The urinary bladder is going to be connected to the kidneys through these ureters. Okay, so the ureters are these little tubes that extend down from the kidneys, and they go down to the renal bladder. So basically, we've got this continuous drip, 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 continuous production of urine. We'll draw this very graphically with little droplets of yellow. The urinary bladder fills up, gets fuller and fuller and fuller and fuller, and eventually when it gets full, you open your urinary sphincter and you pee. Okay, the urine is being produced up here in the kidneys, and the other thing that we want to kind of focus on is the anatomy of the kidney, and we'll be talking about two layers of the kidney. The two layers of the kidney are going to be on the outside. We've got the renal cortex. It's going to the renal cortex is going to be this layer on the outside. Cortex basically means layer on the outside. It's a renal cortex. And the part underneath is going to be, I'll draw it a little bit darker. The part underneath is going to be this kind of striated material. This is going to be the renal medulla. Okay. Medulla. And the space underneath that is the renal pelvis, and the pelvis is where the urine collects before it gets sent down to the kidneys. So down here we'll have the renal pelvis. Okay. And so I want to leave this picture up there for a while because ultimately we'll have to talk about various things that can go wrong with kidney function. But for right now, we want to focus on what's going to happen here, this area right here. Right here in the renal cortex and in the renal medulla, because that's the business end. That's where we're going to be producing our urine. That's where we'll be filtering the blood, and we have to consider the blood flow coming in through the renal artery, and the renal artery kind of breaks up into smaller and smaller blood vessels, and we ultimately will get the blood flowing in through the renal arterioles. Okay, so the blood flow into the kidneys is going to be broken into smaller and smaller blood vessels, into arterioles, which are small arteries. This will be the renal arteriole, and the renal arteriole will end up in this kind of like almost like a blind sac. It's almost like a dead end street. You'll have like this ball of capillaries, and yeah, there's going to be an outlet. There's going to be an outlet. There's going to be a renal, an efferent arteriole. It's going to be carrying blood away from this, but a lot of the volume that's there in the blood, the blood that flows in at pretty high pressure is going to be there's going to be a lot of filtration taking place here. And so there's going to be a lot of impurities, all the impurities that are there in the blood are going to be sent out through the porous walls of the structure. The structure itself is called the glomerulus. And the pressure filtration that occurs is going to send the filtrate into the structure called the Bowman's capsule. That's the way it's described in your textbook. And then from the Bowman's capsule, we'll have some flow through you've got the proximal convoluted tubule, and maybe the convoluted tubule is going to do like a little U-turn here. It's going to come back with a U-turn, and I'm just going to draw it really short. So this will be the proximal convoluted tubule here, and the proximal convoluted tubule is going to send the filtrate down, down, down through the loop of Henle, which is going to extend actually downwards into the renal medulla. Can't draw a straight line here. Okay, so that's the loop of Henle. That's the descending arm of the loop of Henle. And on the ascending arm, it's going to get a little bit fatter. And then we'll have the distal convoluted tubule. We're going to have a little U-turn here. It's going to come back. U-turn here. And then the distal convoluted tubule is ultimately going to empty into the collecting duct. So we'll have a collecting duct that's going to go back down into the renal medulla. And that's pretty much what the kidney looks like. So we'll name these parts. Bowman's capsule we already put on. This will be the proximal convoluted tubule. Distal convoluted tubule, this will be the collecting duct. And this is a long stretch downwards and upwards. This is the loop of Henle. This whole thing. And by the way, this we've actually looked at the two layers that we discussed earlier. It's kind of broken into the renal cortex. And this is a part that's happening up here. This is the cortex. And the renal medulla is down below. So in other words, we're actually taking a cross section right there. And we're saying, yeah, there's going to be lots of renal arterioles feeding into glomeruli, and the glomeruli and the convoluted tubules, renal loop of Henle, tubules, and collecting ducts. So basically these collecting ducts and loops of Henle are the parts of the renal medulla that make the striations, that makes the grain go inside to outside. And all the stuff out here in the cortex would be the renal arterioles, the glomeruli, the distal convoluted tubules, proximal convoluted tubules, etc. Okay, so that's a little bit of the anatomy. Now some of the physiology that's involved here kind of tracks what happens with the process of reabsorption, because you want to remember that the efferent arteriole is going to be carrying blood to the renal capillaries. And the renal capillaries are going to be out here, so this blood is going to be flowing out across the proximal convoluted tubule. Some of the blood is going to be flowing across the distal convoluted tubule. And as it does this, it's going to be picking up stuff. It's going to be picking materials back up. And some of that blood is going to be flowing downwards, downwards, downwards through the renal medulla. There's going to be a blood vessel. It's called the vasa recta. It carries blood all the way down, carries blood all the way back up. And so these renal capillaries are going to be collecting material from the tubules, from the nephron, and ultimately these capillaries are going to go back. And at that point, you will have pretty much served off all the oxygen, so the blood is going to be entirely deoxygenated, and this is the blood that's going to be returning back to the renal vein. So the general scheme that you have been thinking about in the context of our discussions in lectures, that the filtrate passes through the proximal convoluted tubule. It goes down the it goes down the loop of Henle, and it comes back up the ascending arm of the loop of Henle, and stuff is going along the whole way. Here's where we've got the processes of secretion and reabsorption. We get back the things that we want. For example, normally I'll draw glucose in kind of like a light shade of blue. Normally, glucose is one of the molecules that actually gets filtered out. So there's molecules of glucose in here. And we want to get the glucose back. We'll have filtration pushing the glucose out into the Bowman's capsule, but we don't want to pee out the glucose. We want to get the glucose back. So one of the things that happens is that as the stuff is passing through the proximal convoluted tubule, the glucose molecules get reabsorbed back into the blood, and so by the time we actually get to the end of the proximal convoluted tubule, there's no more glucose. And yet the material that flows downward at this point right here is normally glucose free. Okay? No glucose typically. However, if you have an excessive amount of glucose, if we were to overload the system and put so much glucose into the Bowman's capsule at the front end of the glomerulus, by the time we get to the end of the proximal convoluted tubule, there's still going to be glucose. There's still going to be sometimes lots of glucose, and we don't really have any glucose reabsorption taking place after that. So one of the things that we'll see is that if there's a whole lot of glucose in the blood, we're going to be seeing glucose in the urine coming out. because that's one of the things that happens. And if you have glucose in your urine, it makes things a little bit more favorable for various kinds of infections. Um, Yeast infections in particular are going to be something that we find pretty commonly with diabetic patients. And that's mostly because yeasts, being fungi, have a particular fondness for acidic conditions, which is often frequent, but especially sugar. Fungi, like yeasts, are kind of sugar junkies. All right. And so I guess the point here is that we're going to be using urinalysis, or the analysis of urine, to diagnose potential problematic states of the human body. So, for example, glucose present in the urine is an indicator of some type of diabetes. Okay? We're going to be looking for glucose. It's going to be one of the things we'll be looking for. Another thing that we might be noting in a couple of ways is the presence of white blood cells, or pus. Pus is the same as white blood cells we talked about in, I think, the last laboratory. So white blood cells, if you remember, are our infection fighters. So if you have any type of urinary tract infection, we're going to be seeing white blood cells going into the urinary tract to fight that infection, and some of those white blood cells are going to end up, sometimes quite a lot of those white blood cells are going to end up in the urine. And so we'll be running tests to detect pus or white blood cells. Another thing that can go wrong, this is actually pretty serious, is if you were to have any kind of damage in this area right here. Basically, if we were to have some damage in the pressure filtration area, because that's a pretty sensitive spot, because if there's any excessive porosity, if the glomerulus becomes a little bit too porous, and things that are not supposed to flow out from the glomerular basement membrane, if they're flowing out and going into the filtrate, that could be problematic. Normally, we don't have proteins going through. The proteins that come in through the afferent arteriole, they stay inside and they're actually sent out through the efferent arteriole. In some cases, we might actually get some proteins that are filtering out into the filtrate. And so proteins are not supposed to be there because they're normally kept inside the glomerulus. And if we do have proteins, that could be an indication of maybe something that's up with the glomerulus. Maybe you've got some damage to the kidneys, or getting some proteins in your urine through the glomerulus, and that could be a problem. Red blood cells and the breakdown product of hemoglobin are not supposed to be in the urine. The color of urine is supposed to be yellow, not darker. So if you have any kind of like a tea-colored urine, or even worse, reddish-colored urine, that's a signal that you've got some type of hemoglobin leakage or red blood cell leakage. Yeah, and blood or hemoglobin in the urine is generally not a great thing to find. Although it might be something relatively innocuous. For example, if you're on your period, then there could definitely be some blood. But basically, under most circumstances, we don't like finding blood in the urine. And at this point, you might be thinking, well, how much do I really want to know about my health, or how much do I really want to find out about my health in the context of a BIO100 laboratory? And that's a good question. And I guess the bigger question is, well, how qualified is anybody here to make really informed decisions about your health? And that's probably a very legitimate question. And the way that I think about this laboratory is that we're learning about urinalysis. Urinalysis is just a really great non-invasive way of making some, superficial determinations about the state of physiology of the person whose urine is being analyzed. And that happens to be you here. And if you find some concerning result from today's urinalysis laboratory, then maybe this would be a good opportunity for you to follow this up with a visit to your doctor. Okay, so let's go ahead and break down what we're going to be doing with today's laboratory. One is going to be based on the physical appearance and characteristics of the urine. And we'll also have a set of chemical tests, ones that involve test tubes and reagents, some of which are pretty dangerous, so we're going to have our normal safety advisory. And then we're going to back this up with some results that will come directly off of a urinalysis dipstick, sort of a quick and dirty way of getting some results. And finally, we'll be doing a microscopic analysis of urinary sediment. This is a standard test that's run with almost all urinary analyses. And now let's quickly survey the things that we'll be looking or testing for. Just a quick list here. We'll be looking for blood sugar. There's also pus, which are leukocytes, or any signs of infection. We'll be looking for any evidence of blood or hemoglobin. we're looking at the specific gravity of the urine. Specific gravity might vary, and it also might be an indicator of how effective your kidneys are at concentrating the urine. We'll be looking for other things like bilirubin and ketones. And we've got the inorganics like chlorides, sulfates, phosphates. And then we've got a whole set of things that we'll be looking for in the context of a urinary sediment, things like cells, casts, and crystals. And I just want to show you the connections that we're making here. Actually, some things like sugar are being tested for in a couple of ways. For example, we'll be doing a regular chemical test, the normal Benedict's reagent test that we've been doing pretty much all semester long, Benedict's. We also get a read for sugar using the dipstick test. So dipstick and Benedict's are both going to contribute to a call of whether or not we've got sugar in the urine sample. Pus is going to be detected in part by a physical analysis because we'll be looking for cloudiness. And that's going to be backed up by chemical tests. For example, I think we have the nitric acid test to support that. Pus is also going to be tested in using the dipstick analysis. So basically we have multiple ways of determining this. And in fact, even the urinary sediment, because if you have white blood cells, that's going to be showing up in the urinary sediment as well. Okay. Infection, similarly, if you had any kind of bacterial cells or yeast cells in your urine, they would be picked up in the urinary sediment. Blood is being analyzed. We can see the blood is going to be maybe potentially evident in the physical analysis of the urine. We'll be having dipstick tests. The dipstick test will be also allowing us to tell for the presence of blood in the urine. A specific gravity, we'll be using a device called a urinometer, which is basically a hydrometer to test the density of the liquid. And the dipstick test also allows us to detect for the specific gravity or estimate the specific gravity. Bilirubin and ketones, we're only going to be using the dipstick test. The dipstick test is going to be giving us information on bilirubin and ketones. Chemical tests are the only way we'll be able to detect the presence of chlorides and sulfates and phosphates. And the urinary sediment tests will also give us information on cells, casts, and crystals. In other words, we've got all these things that are in kind of a violet color that we're testing for in the urine sample. We've got 4 kind of different methods of testing for these different substances. So in summary, remember that the things that we're testing for here are basically generally considered indicators of the physiological function of things that are happening here, right? You've got many things in different parts of the body that could be going wrong or could be going right, and many of those things will be giving evidence that we could pick up in the urine that's produced. So make sure to come to laboratory with a full bladder. Don't drain the old lizard before coming to lab. We have to have 100 ml of urine in order to run this laboratory. Okay, don't forget. See ya.